Philip Blackman, current president of Engineers and Architects of Hawaii. We invite you to watch our monthly series on Olelo Channel 53. December's Olelo series program is from our November 19th, 2021 Zoom meeting. Our speaker, Dr. George Poore, discusses how prior work on the coronavirus family of viruses in the new mRNA vaccine delivery technique were both critical in the fast development and testing. We encourage your direct participation in this community outreach. So please email us your comments and ideas at eahawaii@gmail.com. Each month, a $50 award will be given for a selected comment or idea on current or past programs.
So this is the humor. It has to do with the younger generation that claims that they are smarter than us old timers. Which stimulated a bit of this rebuttal. Someone has written, if you think you are smarter than the previous generation, 50 years ago, the owner's manual of a car showed you how to adjust the valves. Today, it warns you not to drink the contents of the battery. Now, I actually haven't seen that in my manual, but I go, seems like it's about right. Back to you guys.
Now, Sam Gilly, our secretary, will introduce our speaker, Dr. George Poore.
It is my pleasure to introduce my friend, Dr. Philip George Poore. We've known each other since the first grade at McKinley Elementary School. He attended Stanford, where he studied molecular and cell biology. He received his medical degree from the University of Washington and interned in New York City. He returned to our hometown of Butte, Montana, where he was the chief of surgery. And then he decided that Butte was just too big a city for him and moved to Jackson Hole, Wyoming. But he has kept his finger in the pie on virology and epidemiology. So when I visited him last summer, I asked if he could share with us some of the story of the COVID vaccine and how scientists got the vaccine developed so quickly. I'd heard a news bite that they had designed the vaccine over a weekend in 4 days. He's going to tell us about that, the new family of coronaviruses, and the new vaccines based on mRNA. George, the floor is yours.
So this is a picture of the Tetons on the west edge of Jackson's Hole. Good afternoon, or whatever it is now, I guess. My name is George Poore, and here are some of my bona fides. The most significant thing on this slide, though, is I'm a survivor of 4 years of riding to Butte High School in Sam Gilly's Jeep, which had no brakes. A contrast in timelines. When the Spanish flu killed between 50 and 100 million people at the end of World War I, there was mostly just confusion. George Soper, who chronicled the epidemic, wrote, "The most astonishing thing about the pandemic was the complete mystery that surrounded it. Viruses were essentially unknown." This is a picture of US soldiers that had died in England from the flu. First flu vaccine was not developed until 1945, 27 years later. And the virus that caused the 1918 flu was not characterized until 1990. On December 31st, 2019, the World Health Organization office in China was informed that there was an outbreak of a pneumonia of unknown cause in Wuhan. 4 days later, a metal box arrived at the laboratory of a virologist, Zhang Yongxin, in Shanghai, China. Inside, packed in ice, was a sample taken from a person with pneumonia in Wuhan. After 40 hours of work, he and his colleagues had identified the virus and worked out its genetic sequence. It was a coronavirus. On January 10th, 2020, he published the sequence. The sequence was about 13 pages long. Within 3 days of publication, both Pfizer and Moderna had designed a plan for their vaccine and made arrangements for manufacturing of the mRNA vaccine. How was that even possible? Scientists at Moderna and Pfizer had been working on a platform for rapidly developing vaccines for several years. This platform depended on rapid RNA sequencing, newly discovered ways to make mRNA stable, and newly developed ways to make the RNA delivered into the body. There are several key components to developing this platform. To understand the identity of the infectious agent, to understand how to approach the infection, to understand the genetics of humans and viruses, to understand the mechanisms of human immunity, to be able to deliver a vaccine to stimulate an immune response, to be able to manufacture the vaccine in sufficient quantity and in a timely manner, and finally to get the approval to be able to distribute and administer the vaccine. I will take you through this, which should take about 3 weeks. Before 2020, the average time from the conception of the vaccine to licensure was about 10 to 15 years. The shortest time for the mumps vaccine was about 4. The development of the vaccine for COVID-19 took 11 months from conception to emergency approval. Making vaccines has often been described as a thankless task. In the words of Dr. Bill Foege, nobody ever thanks you for saving them from a disease they didn't know they were going to get. A pandemic is an infectious disease that involves the whole world. This is a graphic depicting pandemics throughout recorded history. Smallpox, this actually talks about the Justinian plague, but I decided I didn't have time to talk about it. But smallpox had a high fatality rate among infants and among adults, but especially in infants. During outbreaks, parents would commonly wait to name their children until after they had survived the disease. A victory for vaccines, smallpox no longer exists in the human population after the last case in 1979. Before antibiotics and vaccine, the primary mechanism of control for ancient plagues was isolation and quarantine. Quarantine came from a Venetian word meaning 40 days, which probably not coincidentally was the amount of time from the exposure to death from the plague. In the 14th century, the Black Plague wiped out an estimated 30% of Europe's population. People suspected of having the disease were forcibly confined in their dwellings, facing both the disease and starvation. Since the 20th century, antibiotics have been very successful against bacterial diseases such as the plague. Antivirals have been less successful. Antivirals mostly only reduce the duration of symptoms of illness in an already infected person. For viruses, vaccines are currently the best defense. Antivirals will also protect you if you take them after exposure and before you get sick. The problem with a disease like COVID is people spread it for several days before they know they have it, so determining exposure is difficult sometimes. In the fall of 2019, the current pandemic began. This was the third coronavirus outbreak in the last 2 decades. There are 7 different coronaviruses known to infect humans. 4 of these cause only colds. The other 3, SARS, MERS, and COVID, lead to severe disease and a fatal outcome in some. The 2002 to 2004 outbreak of SARS infected 8,000 people across 29 countries and had about a 10% mortality. The MERS that emerged in Saudi Arabia in 2012 infected 2,500 and had a 35% mortality. SARS-CoV-2, or COVID, has infected more than 250 million people and has a 2 to 3% mortality, which is about the same as the 1918 flu. The coronavirus develops its name from the crown-like appearance under electron microscope, which you can see here. So these little spikes are called spike proteins. They're what make it look like a crown. These are RNA viruses and involve a single strand of RNA wrapped in a protein capsule made of 4 different proteins. One of these, the spike protein, attaches to cells that have an ACE receptor. The ACE receptor is part of the body's blood pressure system. These receptors are mostly found in the lungs and to a lesser degree in the blood vessels and small intestine. Attachment to these ACE receptors allows the virus to get into the cells. It's really the distribution of the ACE receptors that determines the organ systems attacked by the coronaviruses and the symptoms. Fortunately for the makers of COVID, the COVID vaccine, studies had been done in 2009 comparing proteins on the 2002 SARS virus. These studies discovered that part of the spike protein induced a long-lasting immunity without other undesirable side effects. So they kind of got a head start. To understand viruses, it's important to understand a little bit about genetic material. DNA is made of long sequences of pairs of bases that form the programming language of life. DNA is composed of 2 strands, while RNA only has 1. RNA has one difference in that uracil replaces thymine to match adenine. So this pair is a little bit different in RNA. There are about 6 feet of DNA in each and every human cell. The information in DNA is not used directly. DNA is copied onto mRNA, which stands for messenger RNA. Each base in DNA is matched with a corresponding RNA base. Then the mRNA is fed into molecular machines. These are ribosomes that make the proteins. They're made from the program encoded in the mRNA. Proteins in turn make up the structure of cells and allow the functions of life. In order to decode the viral RNA and create a vaccine, the genetic sequence of the coronavirus had to be read. Genetic sequences were unreadable until 1977, when Frederick Sanger invented a process to sequence bacterial DNA. The process is now in its 3rd generation of automation. Finished in the year 2003, the Human Genome Project took 13 years, over $1 billion, and was about 3.2 billion base pairs. Today, the same can be done in less than a day for under $1,000. Pictured here is a mini-ion. I don't know how you pronounce it, something like that. Using this device to track the virus in real time, that the 2013 epidemic of Ebola virus in West Africa marked the beginning of large-scale, real-time molecular epidemiology. This technology squeezes a single strand of DNA through a micropore and notes the specific electrical charge for each base as it passes through the micropore, reading the code as it goes. A desktop version of the mini-ion can read more than 64 million bases per second. The coronavirus injects its own RNA into our cells to hijack these mechanisms. It turns the infected cell machinery into a virus-making factory. I thought these are little coronaviruses budding off of a cell. That's an electron micrograph. I thought that was a cool picture. And this is a schematic of a coronavirus infecting a cell. The virus uses a receptor on the surface, the ACE receptor, to gain entry into the cell. And the injected RNA is then used to make new virus particles. It uses the ribosome to make proteins, and the proteins help make more mRNA, as well as making new viruses that are then sent back into the body to infect more cells. To combat invading viruses, our body's immune cells develop knowledge about the proteins that make up the virus. Specific cells called antigen presenting cells engulf the virus and present viral proteins to other specialized cells, B cells and T cells. These cells learn and remember each specific virus encountered. The antibody response takes between 5 and 10 days to begin forming antibodies that specifically recognize an invading virus. After that time, B and T cells can recognize and kill invading viruses and cells that are infected with the viruses. The body recognizes a virus by B cells creating antibody proteins. Antibodies have specialized sites which the body has made to fit and bind the viral protein. The antibody and virus then form an immune complex that is transported to where it can be destroyed or deactivated. The next key to understand is to understand vaccination. Inoculation, which was used in the days before vaccine, involved taking material from the smallpox pustule and lancing the skin, depositing the pus below the skin, and then hoping that the resulting infection would not be severe. Mortality for inoculation was about 3%, which is still less than the more than 30% for smallpox in general. Edward Jenner, a doctor in England, noted that milkmaids who frequently got mild cowpox did not get smallpox. He inoculated James Phipps, an 8-year-old boy who was the son of his gardener, with cowpox. Later, he inoculated the boy with smallpox, but smallpox did not develop. Jenner famously wrote a paper to the Royal Society in London describing his experiment. In what was probably the worst editorial decision in the history of medicine, the Royal Society refused to publish it and told him he should stop his experiments. Fortunately, he continued. Vaccines work by tricking the body into thinking that it's being infected. A vaccine presents the body with a part of the infecting virus or an inactivated or weakened virus. Antibodies can then be formed against that protein or vaccine or virus. Once the target of the target protein of the vaccine has been decided upon, the next problem is to getting it into the body and cells. Only then can an antibody response be generated. The developers of coronaviruses vaccines had to choose among 4 options. Injecting a protein directly would seem to be a good answer to the problem, but it tends not to have a very robust immune response. Vaccines using proteins need to add additional adjuvant substances to increase the body's response to the protein. Viral vector vaccines, like the modified adenovirus used by Johnson & Johnson, used the DNA of the virus, of a virus known to mildly infect humans. It delivers the genetic material into the cell nucleus where it is made into the target protein. A downside is that immunity may already be present to previous forms of that virus or similar viruses, and the body may destroy the viral vector vaccine, making it inactivating it. In most attenuated virus vaccines, which are the currently most commonly used vaccines, the virus itself can be changed by multiple passes through a non-human host, like a fertilized egg. This is done with flu vaccines. It then loses the ability to replicate in humans. In some flu vaccines. It's also the measles, mumps, and rubella vaccines are all done this way. And the resulting virus can be given directly to stimulate immunity. This has been a frequently used technique because it does not require specific knowledge of the proteins involved in the virus or its genetics. To produce large amounts of vaccine in this way takes time, though, and there is a risk in those with compromised immune system in that the virus may mutate and cause an infection. The last option and the primary focus is nucleic acid vaccination. DNA vaccination was thought to be the holy grail of vaccines, but it has been proved difficult. And DNA must get into the nucleus, not just into the cell, and it also carries the risk of permanent incorporation into the genome. RNA, on the other hand, does not carry the risk of being incorporated into the germline DNA and does its work outside of the cell's nucleus in the cytoplasm. Historically, the problems with RNA are that unmodified RNA is unstable and rapidly destroyed in cells. Its use also requires the ability to read and write RNA. Injecting DNA and RNA directly into muscle tissue resulted in test animals dying of shock. Finally, there was no reliable delivery method that existed. Normally, RNA introduced into a cell is almost immediately destroyed, but two doctors, Weissman and Carrico, in 2010, and they probably will get the Nobel Prize for that, discovered that a small modification of one of the components of the RNA would allow it to survive and still be able to have its information converted into proteins. This is a kind of marginal drawing that I added to. This discovery made it possible to make RNA vaccines. Since the RNA is fragile and difficult to introduce into the body, another key was the development of a delivery system, the stable lipid nanoparticle. The first time that FDA approved the use of a lipid nanoparticle was a drug delivery system in 2018. Cell walls are made up of two layers of fatty molecules, and the lipid nanoparticles with RNA packaged inside can merge with cell walls to deliver the vaccine mRNA into the human cells. A 2019 discovery that changing the surface ionization of the nanoparticles made them more stable in blood allowed the lipid nanoparticles to be used as a delivery system. The target cells then make the spike protein using the delivered mRNA. A final key was the development of the microfluidic T-junction mixer in 2008. Prior to this, only small amounts could be made at a time using ultrasound emulsification, but this enabled the manufacturing of vaccine in quantities needed to vaccinate the world. Another factor in the rapid approval process had to do with the pandemic itself, with the number of people being at risk for having the disease during the pandemic. If a vaccine is given to a population with no risk of getting the disease, one can't learn if it protects against the disease or not, because the disease is not in the population. My elephant repellent works really well. So having the disease spreading in the population meant that researchers were able to accumulate comparison data between control groups and the vaccine relatively quickly. A disease affecting a much smaller portion of the population would require special techniques, such as tracking down and using contact populations for control and vaccination groups. This type of technique takes a lot longer to accumulate sufficiently large enrollments to demonstrate the effectiveness or ineffectiveness of the vaccine. Large numbers of cases accumulating rapidly allowed this compressed timescale, which the FDA approved. In summary, the steps required to develop a bioplatform allowing a COVID vaccine in record time were, first you had to identify, understand viruses and DNA, and that really started about 1950, and viruses maybe 1930, but RNA needed to be modified to survive in cells and encode the protein. That was 2008. Manufacturing with the T-junction mixer was 2011. DNA sequencing in real time didn't happen until 2015. Stabilized lipid nanoparticles that would stay alive in the blood didn't really happen until 2018. And then the special circumstances of the pandemic allowed for a more rapid approval. Apparently, for this patient, the kill was neither safe nor effective. The difference between the FDA authorization and full approval, Sam asked me to talk a little bit about that. The only real difference is that emergency use authorization is only granted during a public health emergency when there are no adequate approved and available alternatives. And it is only valid when the emergency exists. FDA approval, there's a small group to determine any side effects, a bigger group to try to determine the correct dose, and then thousands of volunteers to determine the safety and efficacy of the vaccine. And then before approval, the manufacturing process is evaluated, and then there are FDA advisory boards that review the data and make a recommendation to the FDA. Quotes of our times. Daniel Patrick Moynihan, who spoke at my graduation, said, everyone is entitled to his own opinion, not his own facts. Kellyanne Conway said Sean Spicer gave alternative facts to that. And Donald Trump said that's fake news. COVID misinformation. People who are dying of COVID are people who would have died anyway. That can be a hard question to identify. If you compare excess deaths, and that's basically comparing the number of people who die on a yearly basis to the current year, you can see that given the last 5 years, the excess deaths from 2020 showed a 14% increase in deaths. Also of note is that the baseline without COVID would have actually fallen. Trauma is the leading cause of death in the US, and trauma admissions were decreased by more than half in the US in 2020. The UC Irvine Trauma Admission Center were down by 80%. Flu deaths were also significantly down. Normally there are 20,000 or more flu deaths in a year, and there were only 646 deaths attributed to the flu in 2020. It's widely stated that Bill Gates developed a nanobot to track vaccine recipients that's in the COVID vaccine. Bill Gates actually did fund research to help avoid preventable deaths in parts of the world where vaccination records aren't available. Many vaccines require multiple doses spaced out at certain intervals, and without accurate records, people may not receive all the necessary doses. So they created an on-patient decentralized medical record, which are microdots that are copper-based quantum dots and are readable for 5 years and injected at the time of the vaccine. This is not used in any COVID vaccines, though. Vaccines don't work. Well, it looks like they do. These are rates of COVID catching the disease by vaccination status. Basically, you're about 6 times more likely to catch the disease if you are unvaccinated compared to vaccinated. And risk of dying, you're about 13 times more likely to die than if you had the vaccine. And that's it. Thank you.
Appreciate it. Everybody
Thank you all for suffering through this.
Very good, very informative. You hit all of the points that I asked for. We have to present you with our certificate of appreciation.
Founded 1902. Wow.
The meeting usually continues, but officially the meeting is over. Those who want to stay on can stay on, and those who need to take off are welcome to leave. And if you can stay on for a few minutes, George,
Sure.
Maybe some people have 1 or 2 questions they might want to ask.
Engineers and architects of Hawaii welcome your comments on this program and any of our recent programs. We encourage your direct participation in this community outreach. So please email us your comments and ideas at eahawaii@gmail.com. Each month, a $50 reward will be given for a selected comment or idea on current or past programs. Mahalo.